So in this video, we're going to be talking about the poisons of oxidative phosphorylation. So we said before that the main goal of the electron transport chain was the process called oxidative phosphorylation. Now, why is it called oxidative phosphorylation? Because there's a process of oxidation that happens between the different complexes that are located on the inner mitochondrial membrane. And there is a process of phosphorylation that happens with the protein ATP synthase, in which it phosphorylizes ADP to synthesize ATP. So these two together are called oxidative phosphorylation. Now, we have three groups of chemicals or drugs that basically inhibit oxidative phosphorylation. The first ones are inhibitors of electron transport, the second are uncoupling agents, and the third are ATP synthase inhibitors, and we're going to be talking in detail about each one in the following video. So we're going to begin by discussing inhibitors, or the inhibition of electron transport. So let's start building our model. So here we have the outer mitochondrial membrane, and here we have the inner mitochondrial membrane. The electron transport chain is going to start with a molecule of NADH that's going to be oxidized by complex 1 to form NAD+. So the NADH electron is going to go to complex 1. Then we're going to have a molecule of FADH2 that's going to be oxidized by complex 2. So FADH2 is going to give its electron to complex 2. Both complex 1 and complex 2 are then going to transport their electrons to coenzyme Q. Coenzyme Q is going to hand it down to complex 3. Complex 3 is going to give it to cytochrome C, and cytochrome C is going to hand it down to cytochrome to complex 4. Complex 4 is then going to donate its electron to oxygen. That's going to react with hydrogen to form water, and this is going to be the last step in electron transport. So each time a complex gains an electron, it's going to enter high energy state, and this is going to allow it to pump hydrogen ions from the mitochondrial matrix into the intermitochondrial space. So after a while of electron transport, we're going to have the accumulation of hydrogen ions in the intermembrane space. And this, of course, is going to create a gradient between the intermembrane space and the mitochondrial matrix. Now, this gradient is going to be used by the enzyme ATP synthase to synthesize ATP. So as hydrogen ions flow from the high concentration in the intermembrane space to low concentration in the mitochondrial matrix, ATP synthase is going to phosphorylate ADP to form ATP. So the inhibitors of electron transport are going to be drugs or chemicals that inhibit these complexes directly. So let's start with the first chemical. The first chemical is called rotin-1, and this is an insecticide. And what rotin-1 is going to do is that it's going to inhibit complex 1. And the inhibition of complex 1 is going to inhibit electron transport. So complex 1 is not going to be able to transport its enzyme to coenzyme Q. So this is going to block electron transport through these complexes. Now, the blockage of electron transport through these complexes, they're not going to enter a high energy state, so they're not going to be able to pump hydrogen ions into the intermembrane space. And if this, of course, is going to decrease the proton gradient. There is going to be a decrease in ATP synthesis because the lower the gradient, the lower the ATP synthase can produce ATP. And there's also going to be a decrease in oxygen consumption because complex 4 is not going to donate its electron to oxygen to form H2O, so oxygen is not going to be needed. The second drug is called antimycin A. So antimycin A used to be an antibiotic that was used. These days it's not used very much, and it's going to inhibit antimycin A, and it's going to lead to the same effects as rotin-1. So it's going to inhibit electron transport, it's going to decrease the proton gradient, decrease ATP synthesis, and decrease O2 consumption. Okay, so now we're going to talk about carbon monoxide and cyanide poisoning. Now, carbon monoxide and cyanide are going to inhibit complex 4. And this, of course, is going to lead to inhibition of electron transport, decrease the proton gradient, decrease in ATP synthesis, and decrease in O2 consumption like we discussed before. But we're going to dive into a little bit more detail about the mechanism of how carbon monoxide and cyanide inhibit the complex 4. So before we get into the details of carbon monoxide poisoning, we need to understand the structure of hemoglobin and also the structure of complex 4. So complex 4 is a cytochrome, meaning that it's going to contain iron in its structure. Now this iron is mainly going to be in the ferric state, or the Fe+3 state. Hemoglobin, as you recall, contains iron in the Fe+2 state, or ferrous iron. Now something important to know about carbon monoxide is going to attach to the iron in the ferrous state, or the Fe+2 state. This means that carbon monoxide is predominantly going to affect hemoglobin, and it's minimally going to affect the complex 4 in our mitochondria. Okay, so when someone gets poisoned with carbon monoxide is going to enter the blood, and it's going to bind to iron in the ferrous state, or the Fe+2 state, and that's going to inhibit oxygen attachment onto hemoglobin. When carbon monoxide reaches our tissue, it's going to bind to the small amount of ferrous iron that is located, and this is of course going to inhibit complex 4 as a whole, and this is going to inhibit electron transport. But remember that the effect of carbon monoxide on the mitochondria or on complex 4 is very minimal compared to its effect on hemoglobin in our red blood cells. The next thing that we're going to discuss is cyanide poisoning. So cyanide is the opposite of carbon monoxide. Cyanide prefers to bind to iron in the Fe+3 state. So cyanide is predominantly going to affect the mitochondria, and it's not going to affect our RBCs. So when someone gets cyanide poisoning, these cyanide molecules are going to make their way all the way to the cells, and they're going to bind to the Fe+3 located on complex 4, and they're going to inhibit complex 4 in general. And this, of course, is going to inhibit electron transport from complex 4 to oxygen to form water. Okay, so what are some important clinical findings of cyanide poisoning? Well, the first thing is that these patients are going to have bright red venous blood. Now, why is that? Well, if you recall, we said that complex 4 donates the electron to oxygen to form H2O. So if complex 4 is inactive, it's not going to need any oxygen. So oxygen is going to remain in the venous blood, and this is why this blood is going to have a bright red appearance to it. These patients can also develop lactic acidosis, and the reason for that is that, like we said before, pyruvate is going to be converted to acetyl-CoA, where it enters the TCA cycle, where it produces NADH and FADH. NADH and FADH are then going to enter the electron transport chain to produce ATP. Now, this process requires oxygen to be present and utilized in order for it to go through. Now, what happens in cyanide poisoning is that when you block the electron transport, you're not going to need oxygen. So the cell is not going to be able to synthesize ATP via this process. So all the pyruvate inside of our cells is going to be shifted to lactate production, and this is why these patients develop lactic acidosis. So, okay, so in the modern day, how do people get cyanide poisoning? Well, the most common clinical scenario is that a patient presents to the emergency room with urgent hypertensive crisis. So he has very high blood pressure. So this patient is going to be started on nitroprusside. Now, the thing about nitroprusside is that it contains, for every 5 molecules of nitroprusside, there's 1 molecule of cyanide. So if a patient is taking nitroprusside for a prolonged period of time, and we're talking here more than a day or two, he can develop cyanide poisoning. So how do we treat this? Well, we tend to give these patients drugs called nitrites. Now, why is that? Nitrites have the ability to go to the hemoglobin inside of our RBCs and convert the iron in our hemoglobin from the ferrous state to the ferric state. So from the Fe+2 state to the Fe+3 state. Now, when hemoglobin is in the Fe+3 state, this is called methemoglobin. Now, all that cyanide that is located in the patient's body is going to bind to the methemoglobin, so it's going to bind to the hemoglobin inside of our blood, and it's not going to affect the mitochondria. And so we do this in order to give time for the cyanide to wash out. So the second groups of poisons of oxidative phosphorylation are called uncoupling agents. Okay, so what are uncoupling agents? So these are agents that are going to cause a hole in the inner mitochondrial membrane. And this hole, of course, is going to cause the leakage of protons from the intermembrane space into the mitochondrial matrix. And in the process, they're going to produce heat that can present as a fever. Now, the reason that they're called uncoupling agents is that they're going to uncouple oxidation from phosphorylation. So oxidation or electron transport that happens in the complexes is going to persist. But because of the low gradient that is produced due to this hole in the inner mitochondrial membrane, there's going to be minimal phosphorylation. So in conclusion, we uncoupled oxidation from phosphorylation. Okay, so like we said before, this leak in protons is going to cause a decrease in the proton gradient. This decrease in proton gradient is going to cause a decrease in ATP because it's the proton gradient that's going to push ATP synthase in order to phosphorylize ADP to form ATP. So this decrease in, okay, so the decrease in the proton gradient is also going to cause an increase in electron transport. And the reason for that is that the electron transport chain is going to try to compensate for this leak by increasing how many proton ions it's pumping into the intermembrane space. And of course, an increase in activity of the electron transport is going to increase O2 consumption. Okay, so we have a few examples of uncoupling agents. The first is called dinitrophenol. Dinitrophenol is used in in the labs as a decoupling agent, and it's just going to cause a hole in the inner mitochondrial membrane and uncouple oxidation from phosphorylation. Clinically, aspirin overdoses is going to cause uncoupling, and this is why patients who have aspirin overdose tend to have a high fever. Now, uncoupling can also happen physiologically. In humans, this is called brown fat. So newborns contain high amounts of brown fat. Now, when brown fat undergoes lipolysis, it's going to release something called uncoupling protein 1, also known as thermogenin. This thermogenin is going to cause a hole in the inner mitochondrial membrane that's going to lead to the leakage of protons, and that's going to produce heat. And this is how newborns maintain their body temperature. Okay, the third groups of poisons for oxidative phosphorylation are called ATP synthase inhibitors. And there's only one example of this, and this is going to be oligomycin A, and this was an antibiotic that was used back in the day. So oligomycin A is going to inhibit ATP synthase, and if you inhibit ATP synthase, you're going to inhibit the process of phosphorylation of ADP to form ATP. So inhibition of ATP synthase, you're going to decrease ATP production. The inhibition of ATP synthase is also going to increase the proton gradient because protons are now going to be trapped in the intermembrane space and they're not going to be pumped through ATP synthase because ATP synthase is going to be inactive. This increase in the proton gradient is going to inhibit complexes, and this is going to stop the electron transport. Okay, so let's illustrate this a bit. So if you inhibit ATP synthase, you're not going to pump the protons from the intermembrane space into the mitochondrial matrix. So all these protons are going to accumulate inside of the intermembrane space, and this is going to inhibit the complexes that are thus going to inhibit electron transport.